Hi, this is Lauren from Cellometer. I am here today to walk you through the unboxing and setup of your Cellometer K2, X2, or Spectrum. Today we will be unboxing the Cellometer X2. This instrument is very simple to set up. Each instrument comes with an AC adapter and power cable, a USB thumb drive with Cellometer software, a quick start guide and user manual, and a starter kit consisting of slides, fluorescent reference beads, and AOPI viability solution. Once we have set up the instrument, we will need to set up the laptop. If you did not purchase a laptop from Cellometer, you will need to provide one in order to run your instrument. Let's plug the instrument in and ensure it powers on. The power switch for this instrument is located on the back above the port hole for the cable. Okay, we are ready to go. As you can see, the instrument is on by the bright white light on the front of the instrument. Once powered on and connected, we can open the Cellometer software. In order to start analyzing any samples, you will need to take a background image. It is also recommended to retake the background image anytime you relocate the instrument. Once the background image has completed, you can go ahead and preview your bright field image. The background preview image should be a bright white image. Cellometer provides a starter kit with this instrument. Let's take a look. It contains the quick start guide and user manual, a box of Cellometer slides, and a vial of fluorescent reference beads. Here are our SD100 slides. They have a plastic film on both sides. You will need to peel off the top and bottom layer in order to pipette your samples. Here we have a 1 milliliter vial of fluorescent beads. Invert the bead vial to ensure proper mixing of the solution before using. Now we can pipette 20 microliters of the beads directly onto the slide using one of the portholes. Insert the slide into the instrument and give the material 5 to 10 seconds to settle. In the software, you'll want to select the following. For K2, you will select the PBMC assay, X2, the yeast AOPI viability assay, and for Spectrum, the S5 AOPI viability assay. Then you can click Preview Bright Field 1. You can adjust the focus using the focus knob on the side of the instrument. See how this image is blurry? We will need to adjust the focus. The beads will be in focus when they display a bright white center and a crisp dark outline similar to that of cells. Then we will preview the green and red fluorescence channels to verify that fluorescent signal is present. Once we have done that, you can click the Count button at the bottom of the screen. After the count has finished, the results screen will appear, and you can close out of the results to view the images on the screen. Go ahead and select the green fluorescent image and check the counted box on the right-hand side of the screen. All of the counted cells will be circled in green, which is displayed on the screen. Let's look at the red fluorescent image as well. The counted cells for this channel will also be outlined in green. Also, we can look at both the fluorescent channels at the same time by clicking the combined box. This will have the green fluorescent cells outlined in green and the red fluorescent cells outlined in red. This was fast and easy. Thank you for watching. Check out the rest of our videos on nexcellometer.com or our YouTube channel.